My name's Maddie Morris. I'm an undergraduate student coming out of the University of Arizona. And this summer I worked with Dr. Oshner at Oklahoma State University. And I also worked very closely with his graduate student, Cole Diggins. So the purpose of the project that I worked on this summer had to do with soil structure. So soil structure is relatively important in natural ecosystems and in croplands, since it can impact root penetration, water infiltration, and water and nutrient absorption by the plants. And oftentimes we can see that soil structure can be disturbed by tillage or by other land management practices, which can also poorly impact soil health. So the issue with soil structure is that it is very difficult to quantify. There are a lot of different physical qualities of soil that combine to just general soil structure. So what we decided to do is investigate a scoring procedure called the Visual Evaluation of Soil Structure, or shorthandly we call VES, created by Ball et al., so and his other contributors out of the UK in 2007. So this process is made to be simple, so to be able to be done by both scientists and farmers and landowners to kind of gauge the soil structural health of cropland soils. It uses a scoring chart, which provides scores from 1 to 5. Generally, you begin at the top, a score of 1, which is the best soil structural quality. And as you go through each of the characteristics, working down, up or down the chart to different scores. So like I said, the objective of this project was to examine, the VES method and see how those scores compare to quantitative laboratory analyses. And then we also, the design of this project was to examine structural differences between cultivated and uncultivated soils. So in order to do that, we found 6 sites across Oklahoma and Kansas and paired a cropland site with a neighboring uncultivated site. And we sampled 12, locations across like both of those paired sites. So 6 in the cultivated site and 6 in the uncultivated sites. And we decided to sample along a line transect so that we could standardize the distance between each of those sampling spots. And at each sampling site, we did a VES scoring method, which would split the block into 2 layers and provide a score for each layer that was then provided a weighted average score for the entire spot that we sampled. And then we also collected soil cores of each layer, each depth. And I don't think I mentioned, but the total depth of the soil blocks we collected were 30 centimeters or less, depending on how shallow the soil layer was where we were at. And these are some pictures of our OK002 site, which is in Chattanooga, Oklahoma. On the left is the soil block from the winter wheat fallow field. And this is the VES chart created by Ball as well. So the green scores are good scores, intermediate and poor scores. And likewise, this is a soil block from the neighboring uncultivated site in Chattanooga, which was a pasture. So for results, this graph on the left, we've plotted the locations of each sampling site or each sample where we took a VES score measurement at each of the 6 sites across Oklahoma and Kansas. So you could see the VES score plotted by the distance at which we took the samples. And some of them we were limited in terms of space. We tried to evenly space them 20 meters apart, but especially in the uncultivated sites, that can be a little variable. And then in this graph on the right here, these are the average or mean structural quality scores that we took from each of the sites. And cultivated is the lighter green color, and we can see just visually that those had higher SQ scores, which indicates a poorer structural quality. And on the other hand, the darker uncultivated bars show, relatively lower or better quality of soil structure. And then also down here, I have a table of just the details about the sites that we sampled to look through as well, and which I'll talk about soil type in terms of our just the results. So we could see visually that there was a almost significant difference between cultivated and uncultivated sites, and we confirmed that with a two-way ANOVA test. And in this case, we had the random effect being the different locations, so the sites in Kansas versus Oklahoma, and our fixed effect was whether it was cultivated or uncultivated land. Which that falls in line with our hypothesis. We kind of did predict that, our cultivated lands due to tillage would see perhaps poor structural quality versus the uncultivated lands. An interesting thing we noted while taking our samples and doing the scoring was that soil water content and clay content, those are very variable in terms of, different soils. So we suspect that those might also impact VEST scores. Across Oklahoma and Kansas, that's not a super grand distance, but if we were taking samples from like across the country, we would have very, very different soils and would suspect that those would also be a variable leading to whatever scoring results that we had. Specifically, we noted that soils with low water content were more compact, so they were harder to break up, and that quality in the VEST scoring method led to a poorer VEST score. Likewise, soils with higher clay content were often denser, which in the same way led us to lead more poorer scorings for those. So we also did have a very wet summer. So we were a little delayed in our soil sampling, but we anticipate completing laboratory analyses with the soil cores we collected. And we are going to analyze the saturated hydraulic conductivity, soil water retention, bulk density, and organic carbon. So once we get those results, we're going to compare those to what the VEST score methods were telling us about the soil structure and see how those compare. And lastly, just overall, our VEST results suggest that, the history of cultivation of certain lands can impact the structural quality, and usually cultivation for agriculture can degrade soil structure quality. And we hope that these results can be a baseline for other land management efforts or other analyses using the VEST method. Lastly, we'd like to just thank the Dr. Patrignani's group at Kansas State for welcoming us and helping us with the Kansas sites. And same goes to the farmers that let us use their fields.
All right, thank you, Maddie. Any questions for Maddie on her research work?